Hello and welcome back. This is topic 2.4 in the IB biology curriculum. So this is basically all about proteins. So if you want to take a moment and look at everything IB expects you to know for this unit, the understanding, the application, the skills, most of it will be covered. Here's the guidance. So first things first, the building blocks of protein are amino acids. And basically you should be able to not only identify an amino acid, but maybe even draw it. So there's a couple of things you need to kind of realize about this. So an amino acid is composed of kind of five main groups. On the left here we have the amine group, which is always going to be two hydrogens and a nitrogen. It can also be written as NH2. On the right here, we have a carboxyl group, which can also be written as COH. And basically, these two are kind of like the defining units. In every single amino acid, this will be the same. The same thing for this H down here, and as well as just this C. So basically, these are kind of the four main components that are the same in every amino acid. So this is basically how you're going to be able to identify amino acid if you're ever given a picture of it. Now, the differing factor here is this R group. So now, when it comes to the R groups, these are going to vary between amino acids. So there's about 20 different amino acids that create all the different proteins that we use that form life. And basically, IB doesn't expect you to know all these different amino acids or all the different R groups, but you should know that this is the variable that changes the different amino acids. And this is actually what changes protein structure, which we'll talk about a little bit later. But basically, for now, what you need to know is the R group can be polar, it can be non-polar, it can have a positive charge, it can have a negative charge. And basically, every single amino acid is coded by a different genetic sequence of bases, but that'll be covered in a later topic when it comes to the central dogma of biology. So from here, we have to start understanding how we put these amino acids together in order to create these peptide chains, polypeptide chains or proteins. And what we do is we create what's called peptide bonds. So this is basically a bond that is created between two amino acids. Now, this is a anabolic reaction we're building, and it's also a dehydration reaction, a dehydration synthesis, because what's happening here is we're taking a hydrogen and an oxygen and then another hydrogen, and we are forming a water. So if you notice here, we have the amine group, and then the amine group still isn't lost. It's kind of changed a little bit. We're getting rid of that hydrogen in order to make that water down here. And then on the end, we still have that carboxyl group, and if you notice, we'll have those R groups. So notice that we can still find the components of an amino acid throughout this polypeptide chain. It's just now we have a connecting factor, which is a polypeptide bond. So from here, here's an example question if you want to take a moment and pause and answer. So, what molecular structure correctly illustrates two amino acids linked by a peptide bond? Okay, so first thing you can kind of go in to eliminate some answer choices is what you'll have to see is you can tell right off the bat that it's end to end, end to end. It's never going to be two carboxyl groups together or two amine groups together. So these two have amine groups on the ends and carboxyl groups on the ends, actually. They have carboxyl groups on the ends. And what this is telling you is that in the middle here, you're having carboxyl to carboxyl or amine to amine, and this is a big fat no-no. We have carboxyl to amine. It's always carboxyl to amine. So then between these two, we can kind of start looking. So we have that amine group, we have that C. In this case, this is our R group. We have that carboxyl. And notice we have a full carboxyl group here. Now, if we go back, you'll notice that we gave up a hydrogen and oxygen from that carboxyl group in order to make that peptide bond. So this is incorrect, and that leaves us with C. So then we start talking about the bigger structure of the proteins, because the polypeptide chain alone is not a protein. So there's four different layers of structure, and the first one is that is called the primary structure. And what primary structure is it's basically just the order of the amino acids. It's basically how the amino acids line up and how they all connect together. So basically, you'll see here some different examples of different amino acids, and that's basically all it is. It's just a long, super long polypeptide chain. And of course, we'll talk about what determines this primary structure is DNA and how that's transcribed and all that different stuff. But this is basically, for this unit, we're only talking about that protein structure. So the next the next the next is the secondary structure, and this is when we start seeing proteins kind of take shape. So depending on the type of interactions between the different hydrogen bonds, we're going to have either an alpha helix or a beta pleated sheets. And you can notice here that these are going opposite directions, and actually these ones are going opposite directions as well, but sometimes you'll have parallel. So these two right here are anti-parallel, these like blue, these beta pleated sheets, but sometimes they can even be parallel. So if I go back for a second, you'll notice that these amino acids have hydrogens, and they also have oxygens and nitrogens. All of these have the ability to partake in hydrogen bonding. So if we go back here, when hydrogens align like this, we're going to get that alpha helix structure because it's going to start spiraling. But if I kind of make this more beta pleated, it looks more like a folded piece of paper, and we see hydrogen bonds kind of drawn between them. And you don't really need to know the specifics of this, but basically what you're expected to know is that it's these hydrogen bonds that are basically determining the shape of the secondary structure. So then from there we get larger and we get the tertiary structure. So now remember when I said that those R groups can be polar, positive, nonpolar, negative. The specific charges or polarity of these R groups are going to change the tertiary structure. So the tertiary structure is basically the 3D structure, how these proteins start folding together. So say this part was negative here and this part was positive, this is going to kind of come together. And say maybe this part is nonpolar and is hydrophobic, and this part out here is hydrophilic. So this is going to go to the outside, and this is going to want to stay in the inside. Again, you're not going to know the super detailed specifics, but basically know that it's those R group interactions between R groups themselves, as seen between this here, the negative and positive charge, as well as R groups in the environment, as seen between the hydrophobic and hydrophilic R groups that kind of determine this 3D structure. So this final structure is the quaternary structure, and the quaternary structure is just the interaction between multiple polypeptide chains. So if you'll notice here, we have 1, 2, 3, 4 different polypeptide chains, and basically these coming together to create that final protein. This is when we finally start having that protein. At the same time, it's worth noting that there are prosthetic groups. So this protein is actually hemoglobin from the blood, and there are compounds, these little green things, known as hamine, and this is kind of what gives it that oxygen binding properties. And these are not coded by DNA in any way. They are not. There's no way the cells don't make this through amino acids. So these are kind of non-amino acid molecules that are incorporated into the protein, and this is a defining characteristic of that quaternary structure. So here, there's just kind of like some differences between different quaternary structures. There's two main kind of quaternary structures. We have fibrous, which is kind of just like long and narrow, if you think spider silk, and we have globular, which is kind of hemoglobin, which is kind of like roundish. So basically, so those fibrous are long and narrow. Those globular proteins are round and spherical. So basically, you kind of need to know some of the differences between them. So fibrous are going to be more structural. If you notice, like spider silk, if you start looking into muscle, these are all like fibrous proteins, and basically what they're doing is they're kind of providing support. If you They also have a repetitive amino acid sequence, so it'll be that fibrous is kind of just like repetitive amino acids over and over again. It's worth noting that they're less sensitive to changes, which kind of makes sense, right? If they're structural, they have to like be durable. They have to withstand those changes. And then here are just some examples: collagen, myosin, fibrin. You will need to know some named examples, but you will learn more about these throughout the rest of the course. So you don't really need to focus on them right now, but you should start memorizing some examples. Fibrin helps with clotting. Myosin and actin are both help with muscles. Collagen, keratin, all these different things. So and then another last thing, generally they're insoluble in water. So for one example, fibrin is an insoluble protein, structural. So when it helps clot, fibrin comes out of solution and forms that clot out of the blood. So then when we get to globular, this is going to be more your enzyme stuff. This is going to do function. It's going to have serve a purpose. So it's an irregular amino acid sequence, so the structure is not like uniform, it's not repetitive. It's more sensitive to changes. So basically just think the opposite of that fibrous. So we have enzymes, hemoglobin, insulin. They're all serve a purpose. And basically these are going to be the soluble in water. So again, if you think back to that blood, that hemoglobin is going to be able to dissolve in that blood. So finally, as we're talking about durability, proteins will denature, which basically means they will start to lose their structure at certain conditions. So the two main conditions you want to kind of keep your eyes out for are temperature and pH. So basically, these protein structure all relies on intermolecular forces. We have hydrogen bonding between hydrogen and oxygen and nitrogen. So there's a hydrogen bond. This is the strongest intermolecular force. At the same time, we have positive and negative charge. We have polarities. These are all different types of intermolecular forces, which are going to keep these things keep these proteins together. And what happens is, as you get, say, one example for heat, when you start increasing the temperature, what's going to happen is there's going to be more and more energy. And the more energy that there is, think about a gas. It starts getting, it's very random, it's very wild, there's lots of movement. So what happens is these intermolecular forces are broken. So when you break those negative, those positive negative if you just break the intermolecular forces, what's going to start happening is that protein is going to start to unfold. So you go from the quaternary structure, and then it'll start unfolding. You'll go to the tertiary structure, it'll continue to unfold, and then you'll eventually get to that secondary structure. And if it gets to a certain point, those hydrogen bonds will finally break, and you get back down reduced to that first structure. Now you will never see it to where temperature gets so bad that it starts breaking apart the peptide bonds. Those peptide bonds are not going to be able to break between those are intermolecular bonds. Basically, those are the forces holding together the molecule itself, not forces together holding molecules. So this is kind of the stopping point, that primary structure. And actually, it'll be kind of hard to get past the secondary structure since hydrogen bonds are so strong. But basically, just know that temperature will start breaking those bonds. At the same time, if you start, say, each enzyme has a specific pH, because pH affects kind of like it will affect those polarities and how it interacts with the environment around it. So, say we have that pH of 7. If we start getting too high, or if we start getting too low, what's going to happen is those intermolecular forces are going to be messed up again. That polarity is going to change. So basically, maybe something on the outside is going to want to be there at pH of 7, but once you reach a pH of 14, it's going to want to go to the inside. And now you had a protein, or I'd say an enzyme that was functioning, but now that moving portion of the protein changed it, and now that enzyme no longer functions. So basically, from this, you just have to remember temperature and pH. If they get out without those, if temperature gets too high, the protein will denature. If pH gets out of the scale, it'll denature. So finally, we have the proteome. So if you remember, the genome is a collection of all the genes. And then from that, we get that proteome. So the proteome is just basically a collection of every single protein that that a person or an organism can make. And it's worth noting that the proteome will always be larger than the genome, because the genome can be altered. So say we go from DNA to RNA, they can be altered afterwards. We can kind of start changing the DNA after we get that mRNA, after we copy it and start making changes to it. And basically, this is why the proteome is so much larger than that genome. And also, basically from here, we can start talking about the different functions of proteins. IB will also want you to know the different functions of the proteins. So I'll go through them real quick, but you're going to just basically, it's kind of a memorization game. You're going to have to start kind of identifying which different what the different protein functions are. But I will tell you that as you go through the year, you will start seeing more of these over time, and basically it won't be so much as memorization, but finally like learning their role in the body. So we have transportation. If you think hemoglobin, this is going to kind of carry oxygen throughout the blood. If we have for hormones or communicators, we have insulin, and this is going to regulate that blood sugar. So if the blood sugar is too high, it's going to communicate throughout the body. We're going to have to lower that blood sugar. We have enzymes, Rubisco, when you get to plant, or SL won't get to plants, but HL will. But basically, Rubisco is a protein that or is an enzyme that helps with photosynthesis. We have structure, which we kind of talked about with fibrous. We have collagen, which is you should know that this is an important fact here. It's the most abundant animal protein. If you look at the human body, the most abundant protein you're going to find is that collagen. We have spider silk as well, which is actually stronger than Kevlar, as I noted here, and basically that's going to be that structural stuff. We have immunity. When you get to the immune system in topic 6, you're going to see immunoglobulins, which kind of have antibodies that target specific antigens. We have actin and myosin, which are for movement, if you think, because both actin and myosin are for muscles. So basically those muscles will kind of contract, and what will happen is that'll cause movement. And finally, we have sensation. Basically, sorry, I cannot pronounce this, but this rhodopsin, this is all used in photoreceptors. So this is found in the human eye. My kind of drawing of an eye there. So basically, all the underlined ones are kind of named examples that IB kind of wants you to know, and then these, there wasn't really any for movement, but I included them, actin and myosin, just to give you an example there. So now on to a question that you might be asked in the paper 2 for your response section. So distinguish between fibrous and globular proteins with reference to one example of each type of protein. So going back to thinking of that chart, you're going to want to start naming different examples. So of course we have fibrous, it's long and narrow, it's spider silk, and so it's structural. If it's structural, it's kind of uniform, it's repetitive, it's going to have that repetitive amino acid sequence. If it's structural, it has to be durable, so it's less sensitive to changes. And then you'll recall, we have the myosin, the actin for muscles, we have fibrin for blood clotting, collagen, the most abundant protein in the human body, and they're insoluble in water. So basically, and then you kind of think the opposite for globular. For globular, we have the round spherical, it's functional. We have the hormones, and basically they're more sensitive because they don't really need to be durable. They're just communicating. They have a regular amino acid sequence. Because they have this like round shape. It's basically kind of random, right? And then we just think like don't really say enzymes because they want like a named example, but hemoglobin, insulin, immunoglobulin. All of these things will be learned in topic 6, so don't really worry about getting the names down yet, but start trying to remember those names. And then generally soluble in water. So list 3 general functions of non-membrane proteins. So this is kind of getting back to that function. So you start thinking, what are some proteins we know? We know fibrin, which is blood clotting. So that's kind of like protection, structural. We know that rhodopsin, I'm sorry, I cannot pronounce it, but that's basically for like light perception, or it's photoreceptors. We know, and it kind of wants the general, so you don't have to go so much into detail. So we can list those again, those transportation, communication, enzymes, structural, immunity, movement, sensation, all you can just basically list this, but it's worth giving a name example so they really know what you're talking about. So finally, we have explained the primary structures of tertiary, or explain primary structures and tertiary structures in enzyme. So while we really didn't cover enzymes too much here, it's the same thing. Enzymes are proteins, so explain the primary structures. So again, that primary structure is the order of amino acids linked together by peptide bonds, while the tertiary structure is the 3D structure, which arises from the interactions between R groups of the primary structure. So again, we have the chain of amino acids, peptide bonds. We have the folding of the chains into a 3D structure, and basically that's all you need to get those 3 points.